Ooh, already 75 people have joined. Kicking right up. So for those of you that have joined, we're just gonna wait a few minutes to let everybody trickle in, and we'll start in a couple minutes. Okay, well, it's 11:02, so I think we're gonna get started. So I'm happy to welcome everyone today to the Rising Stars in Reproductive Biology webinar series put on by the Society for the Study of Reproduction. As many of you may know, the goal of this series is to showcase the research of new investigators making an impact in the field. And today's speakers, Dr. Farnes Amargant Ivriera and Dr. Kara McKinley, are certainly doing impactful and novel work. So we're excited to have them. Before we begin, though, I just want to give a quick note about what the format of the seminar will be. So today, each speaker will give around a 20-minute presentation, followed by questions and discussion for about a 10-minute period. And I want to remind everyone as they have questions, just they can post them in the Q&A section, and we will go over those afterwards. So with that, I'm just going to jump in by introducing our first speaker. So we're going to start off today with Dr. Farnes Amargant Ivriera, who is an assistant professor in the Department of Obstetrics and Gynecology at Washington University, the School of Medicine in St. Louis. Her research is focused on ovarian physiology and reproductive aging, with a particular emphasis on the mechanical properties of the ovarian microenvironment and its influence on follicular genesis and oocyte quality. We're very excited for her talk. So, Farnes, please take it away.
So, um, thank you very much, Andrew, for the introduction, and thank you to the SSR for giving me the opportunity to present or introduce the research that my lab is doing. So I started my lab almost 2 years and a half ago at WashU Medicine, and I thought that today I can give you an introduction of the research questions that we are addressing, and I will also give a snapshot of 3 different stories, one that was published very recently, and 2 that we are currently developing in the lab. Okay, so my lab particularly studies what mechanisms contribute to equality, and we are addressing this question by analyzing oocyte competence and how oocyte competence is established in the ovary long before the process of ovulation. So all of you know that the follicle is a functional unit of the ovary, and it's composed of 3 different cell types, the oocyte, which is the germ cell, which is surrounded by different somatic cells, the granulosa cells, as well as the theca cells. These follicles are formed before we are born in the ovary, and they are arrested in the follicle, in the primordial follicle. So this is our ovarian reserve, and a few of these follicles will be activated and will start the process of folliculogenesis. So these primordial follicles, the oocyte will grow in size, the granulosa cells will transition to different morphology, the granulosa cells will keep proliferating until the process of ovulation. And there are a few papers from the late 80s and early 70s that suggested that in humans, it can take up to 290 days from the moment in which a primordial follicle becomes activated to the process of ovulation. So this is an extremely long process. As I mentioned, the follicles are assembled before birth, and we have more than 1 million follicles assembled in the human ovary, but actually very few of them will reach a process of ovulation. It's estimated between 400 and 500. So most of the other follicles will die through a process of atresia. So in the lab, we like to think about folliculogenesis as an evolutionary conserved process of follicle selection. So during folliculogenesis, we have several checkpoints that will allow only the highest quality oocytes to keep developing until they reach ovulation, although we know that in some cases, these checkpoints fail. So what are these checkpoints that we have in the ovary? And this is a very general idea, but we have the hormonal regulation. We know that LH and FSH regulates folliculogenesis. We know that there is also this bidirectional communication in the follicle. So the oocyte can secrete proteins like the GDF9, BMP15, then regulates granulosa cells and T cells, and this responds by producing and providing nutrients to the oocyte to continue growing. But it's important that we remember that these follicles, they don't grow in isolation. These follicles, they grow in the ovary. And in the ovary, these follicles go through this process of growing. So the antral follicle is 1,000 times bigger than the primordial follicle. We also have to rupture the tissue during ovulation, and we have to wound heal the tissue. So all these processes, highly dynamic processes, we believe that they regulate, they create forces in the tissue that will regulate the process of folliculogenesis. So mechanical regulation might be an extra layer of process to regulate the process of folliculogenesis and oocyte quality. But why is it important to specifically study mechanical control of follicle development and oocyte quality? And this is data that I obtained from the World Health Organization and the Pew Research Center, where they show that in the US, 1 in 10 women receive fertility services. And these women seek fertility service for many different topics. It can be genetic factors, endometriosis, uterine fibroids, PCOS. But in the last decade, there is one factor that has increased significantly the number of women that look for infertility services, with age aging. So we know that with age, there is a decline of the number of follicles, and there is also an increase of the poor and the fraction of the poor quality of the oocytes. So we have less follicles, and the oocytes remaining in the ovary, they are of less quality. So women were delaying childbearing, and this is, right now is a very important clinical factor, understanding how aging impacts oocyte quality. But why specifically mechanical regulation, right? So this really started when I was a postdoc in Francesca Duncan's lab at Northwestern, and we isolated ovaries from reproductively young and reproductively old mice, and we performed nanoindentation. That is a technique that basically you indent the ovary at different positions, and the force that you need to indent this ovary is a readout of tissue stiffness. So we performed these experiments, and we found that the ovaries from reproductively old mice, are significantly stiffer than the young ovaries. And we knew that this was triggered by this increase of fibrosis in the ovarian stroma. So there is this increase of collagen that triggers this increase of stiffness in the mouse ovary with advanced maternal age. But more importantly, we show that this is a conserved phenotype, because when we analyze collagen composition in the human ovaries, we also saw that increases with age. And the lab of Christiane Amorim in Belgium, they nicely show that the stiffness of the ovaries obtained from menopausal women, they are also stiffer compared to reproductive age women. So at this point, we knew that the stiffness was a mechanism that was happening in the ovary with age. But we also realized that there are many other pathologies in which ovarian function is altered, that they also have this same phenotype, where there is an increase of ovarian stiffness. And probably the most well-known one is the PCOS. This is a human ovary that has PCOS, and the collagen is a stain. You can visualize the collagen in blue. So we have this increase of collagen, this increase of stiffness. The same when we irradiated ovaries, we see that these ovaries become fibrotic and become stiff. So this concept of ovarian stiffness, it's observed in many ovarian-associated pathologies. So we really want to understand how these changes in the mechanical properties, how the mechanical control of the ovary and in follicles impact or regulates oocyte quality. So when I moved to WashU 2 years ago, the first question that we wanted to address is this important? Like, what is the impact that we change the mechanical properties of the ovary? So what are these consequences of increased ovarian stiffness? And we took advantage of the in vitro follicle culture developed in Teresa Boudreau's lab, where we can isolate these follicles. We encapsulate them with alginate, so we can keep the 3D morphology of these follicles. And then we can culture them for 14 days in culture, where we can see how these follicles are growing and we can induce ovulation. So since we have to encapsulate these follicles with alginates, we took advantage of this to generate gels that can mimic the stiffness of the young and the old ovary. So for the first time, we knew the exact properties of the mouse ovary. So we found that the 0.5% alginate was mimicking the environment of the young ovary, and the 2% was mimicking the stiffness of the old ovary. So now we have a system that we can analyze how these mechanical properties impact folliculogenesis without other aging-related factors. So the first thing we did was to do a short-term culture and perform RNA-seq to know if these follicles can even detect this environment and respond to this environment at the transcriptional level. And to our surprise, what we found is that follicles are highly mechanosensitive. They respond to these mechanical cues in less than 24 hours. And we found several genes that are related to mechanosensing or mechanoresponsive that are more abundant in the follicles that are cultured in the stiff environments compared to the 0.5. So these follicles have the machinery to detect and integrate these mechanical cues from the environment. We then analyze what are the pathways that are more expressed. And again, we have another surprise here because we found that the most upregulated pathways in the follicles cultured in the old environment are genes related to inflammation and fibrosis. So I say that it's surprising because we usually think about inflammation as being the first step, and then we generate the stiffness, so the fibrosis and the stiffness. But here, what we can see is that even if you put a follicle in the stiff environment, that it's not inflammatory, it suddenly becomes inflammatory. So it can also happen at the other way around. This was a very short-term culture, so it was only 24 hours. So we decided to validate these findings at the protein level during a long-term culture. So we validated using cytokine arrays that we have this inflammatory phenotype. And we did exactly the same for the collagen production. We cultured follicles up to 8 days and we stained them for collagen. And we saw that the 2%, they maintain these high levels of collagen, while the 0.5, there is a decline in collagen content. So this really shows us that this microenvironmental stiffness can trigger a fibroinflammatory profile in the follicles, resembling that of the H mice. So H mice, the follicles are fibroinflammatory. So just changing this environment, you have the same phenotype. What happens at long-term culture if we culture these follicles for 14 days during all the process of folliculogenesis in this stiff environment? So I will show you just a couple of results. So in the first row, you can see here the same follicle that's been cultured in the soft environment for 10 days. And you can see how the follicle is growing and clearly expands between day 8 and day 10. But if a follicle is cultured under 2%, we don't see any differences until day 6. Up to day 6, they look morphologically the same, and the quantifications of the size, there are no differences. But it's at day 8 and day 10, that is when these follicles expand, that they are encapsulated in this compressive environment so that they cannot grow. And this is exactly the same that we see in the H ovary, where these follicles are smaller than in the follicles that we have in the young ovary. We decided to isolate the oocytes at day 12, the last day of the culture, And we found that in the follicles cultured in the STIV environments, more than 90% of these oocytes are completely degenerated compared to only 25% in the follicles from the young, from the soft environment. So this really shows us that this increase of stiffness can impair follicle development. So in this first story of the lab, what we wanted to show is that in a physiologic condition, the ovary is a very soft tissue and it has a very homogeneous environment. But in conditions such as aging or PCOS, these mechanical properties change. If you take a follicle that is a high quality follicle from a young ovary and you culture them in a soft environment that it resembles the young ovary, you have a physiologic phenotype. So follicles are growing, the oocyte quality is high, and you have hormone production. But if you change the environment, you take the same follicle and you put it in a stiff environment that resembles the old phenotype, you suddenly have an aging phenotype. These follicles grow less, the oocyte quality declines, and you also generate this fibroinflammatory phenotype. So in my previous work, I mentioned I measured the mechanical properties of the ovary, but these were bulk analysis, right, of the whole ovary. And we also introduced, I introduced in the first slides that the ovary is a highly dynamic tissue because we have the follicles that grow significantly. There is the process of ovulation and wound healing. And there is also multiple compartments in the ovary. We have the follicles, but we also have the corpora lutea, the stroma, and different cell types. And all these different cell types, they require different mechanical cues to maintain their proper function. So thinking about these dynamics, this really to me resembles this concept in architecture where it says the form follows function. And this was coined by the architect Louis Sullivan in 1986. And it says that the shape, the size, and also the appearance of a building should be guided primarily by the function of the building, by its utility. So I think the same concept can be applied to the ovary, where the biomechanics also can help shaping the function of the ovary. So understanding how this biomechanics shape the function of the ovary can really help to understand better how the ovary works. And in this area, we have so many open questions in the field. Some of them, I put them here. So what are the specific mechanical properties in the different compartments? How the tissue stiffness changes in an aging continuum? Other mechanical properties, not only stiffness. An open question in the field that is the properties of the ovarian cortex versus the medulla, or how these spatial mechanics drive age-associated inflammatory phenotypes. However, to answer these questions, we need to build new tools, and this is what we have been doing in the lab. Recently, we have been working on building a high-resolution multi-scale approach where we can map ovarian mechanics with the structure and the function of the ovary. And this is something we have been doing with Christine Meyer's lab in Columbia, together with Daniela, that she's a PhD student in her lab. So how we build this system? So we isolate the mouse ovaries and we mount them in these glasses where we put some orientation marks so we can always keep the same orientation of the ovary through the different stages of analysis. The first stage of analysis is the nanoindentation. So we mount the ovaries in this nanoindenter that is exactly the same that I showed you at the beginning when we did the bulk analysis. And we can set up coordinates using this nanoindenter. And we indent the tissue different times, creating this map, and each point is separated by 200 microns. So we can build this map on the tissue. Once we have this map built, we perform imaging. So we use optical scope to take an image of this ovary, and then we use optical coherence tomography. And this step is very important because what this microscope allowed us to do is to image the ovary before fixing, and we can take different stacks and we can visualize without any kind of staining the different structures of the ovary. So we can differentiate between corpora lutea and follicles. And once this is everything done, we fix the ovary in the same dish and we mount it for histology analysis when we can identify the different cell types. So knowing that we keep the same orientation and we have the coordinates during the nanoindentation, what we can do now is to align all this information and create this 3D reconstitution map where we can match the mechanical properties with ovarian structure and hopefully get more information about ovarian function. So the first thing that we wanted to do with our system was to analyze the aging continuum. If with aging, like we have the ovaries that are stiffer, but if this is like a continuum mechanism. And we indented ovaries from mice that are 7 to 9 months. That it's when we start to see this fibrosis, this decline of fertility start to happen. And we saw that the increase of stiffness is a progressive increase in the ovary. And just an interesting observation we found is that it looks like the left ovary is always stiffer than the right in the old mice. And this is something that we are trying to understand how is this happening. But one of the questions I mentioned at the beginning that we are very interested to answer is what are the mechanical properties of the cortex and the medulla? Because we have this concept in the field that in the humans, but also in the mouse, the cortex is stiffer than the medulla. And in the human, we have a very big region of collagen enriched region in the cortex, but in the mouse, it's not so evident. So we wanted to know if this is exactly like that way in the mouse. So what we did is to isolate these ovaries, we encapsulate them with agar, and then with a vibrotome, we slice these ovaries so we can have two portions. One portion is the half external, so we can indent the cortex from the external part. And the other portion we use to indent the medulla, the internal parts of the ovary that now is exposed after the slicing. So this is still ongoing. We still don't have all the data to show you, but we hope that with this system, we really can answer the mechanical properties of the cortex and the medulla. But what I want to show you is some preliminary data that we obtained using this mapping in the medulla. And here you can see the maps from the medulla, where you can see that in the young ovary, it's a very homogeneous tissue. The tissue is extremely soft. But when we indented the mid-age ovaries, we start to see that the medulla, some regions become more stiff, while others keep this extremely soft environment. So now we are doing the mapping, so to know exactly which regions are the ones that become stiff. But we have some hypotheses and some data that suggests that this might be like some of these foci of collagen that we see in the ovary. So this is a staining of ovaries from this is mouse and this is human. And as you can see, this is a PSR, so collagen is in red. So in the young mouse, we have very low levels of collagen. But at 7 months of age is when we start to see these foci of collagen that appear in the ovary. And we think that these foci are the ones that are driving this increase of stiffness, these regions that are very stiff, compared to other regions where in the ovary we don't have these foci and remain extremely soft in the ovary. By 18 months, we have that all the collagen is more abundant, so all the ovary becomes significantly stiffer. And similar in the human, we have these foci of collagen that are very regional, so they are very spatially in the ovary. So why we think this is important? Because if you isolate follicles from a reproductively mid-age, this is the case, we isolated follicles, morphologically, they look very similar. But if you do transcriptomic analysis, we know that some follicles, we have a transcriptome that is more similar to a reproductively young mouse. So they express genes related to development, metabolism. But others look very similar, but they have a transcriptome more similar to an old mouse. And this is characterized by this inflammatory profile. So what we think and we are exploring right now in the lab is that maybe these follicles that they have this old-like phenotype where they have an increase of inflammatory markers is because they have been growing very close to these regions of collagen foci, while the ones that preserve this young phenotype, it's because they have been growing in these regions where we don't have this collagen and we still have a soft environment. So we are trying to demonstrate that the environment in which these follicles grow really shapes their potential. So the summary of what we are doing for this project is to build that we have built this system where we can test the ovarian mechanics, the structure, as well as function. That stiffness progressively increases with aging. Spatial mechanical heterogeneity emerges with age, and we are doing these maps to understand the cortex and the medulla and this concept of inflammatory stiff foci and inflammation of the follicles. So in the last minutes, I just want to show you a project that we have that has a more translational perspective, where we started to wonder in the lab, can we use biomechanics to improve IVF techniques? And we know that the use of ART, assisted reproductive technologies, has increased significantly in the past decades, but success rates are still at 30%. So in the lab, can we use oocyte biomechanics to develop a cost-effective system to maximize success rates for ART patients? And this work, it really has been pioneered, and we have been very inspired by the work that Janice Evans started in her lab in the mice, using mice, as well as the work from Marie-Hélène Belrac and Marie-Émile Terret, where they really show that the cortex, the mechanical properties of the cortex of the ovary, of the follicle, are very important regulators of oocyte quality in the mouse. For example, here we see that the mechanical properties of the oocyte cortex decline with maturation. And this is an example of when they created a mouse model with a cortex that was softer, and they saw that this induces problems in the spindle positioning as well as increase of aneuploidies. So in a typical IVF procedure, the women get stimulated with like the hormonal stimulations, and we usually obtain a mixed population of oocytes. The clinic, they use the oocytes that are mature, the XM2 oocytes, while the ones that are arrested at the germinal vesicle, the mature ones are usually discarded. And although we have systems to mature these ones to M2s. And this is a project we are doing with the REI division here at WashU. And we thought, in the clinic, most they don't use the GVs because they don't know if these GVs can mature to an euploid oocyte that will eventually become blastocyst. So maybe this system to screen these oocytes, GV oocytes, might not be important for all the patients, but we think there are 3 populations of patients that will really benefit from having this system to screen GV oocytes for their potential to mature to high quality oocytes. And these are patients that have high number of GV oocytes, so 50% of the retrieval oocytes are GVs. They overall have a low number of oocytes, or they can only go through one ovarian stimulation. So for all these patients, having one more mature oocyte, it's really important and can change the success rates of their clinic. So what we are doing is to get these oocytes from the clinic. We perform this aspiration, that is the same system that Janice Evans was doing she's doing in the lab, where she aspirates the oocytes to measure the mechanical properties. This is not invasive, that's why we like a lot this system. And then we allow them to mature, and we correlate the mechanical properties with the maturation as well as oocyte quality using subcellular analysis. And in parallel, we are developing an AI-powered predictive system with Peinan Zhao and Matt Versi, where we can use the information that we get from these videos of how the oocyte deforms to provide more data to our model. So this is how it looks like in our system in the lab. So it's mounted in an ICSI system. So the only thing that you need is an aspiration pump that is $5,000. It's very fast. You do the aspirations in less than 2 minutes. And any embryologist that is an ICSI trained can do this procedure. So we have very few preliminary data, but we are very excited. First thing we show is that during aspiration, we don't see changes in the maturation rates. So the oocytes are not impacted by this aspiration. This has been demonstrated in the mouse, but we also wanted to show it in the humans. And this small sample size of 30 oocytes that we have with 10 patients, we see that there are different mechanical properties for the oocytes that can mature to an egg, or the oocytes that rest in the GV stage. We even ran some preliminary tests using a ROC curves, and we saw that the area under the curve is 0.7. So we are very excited to continue this work, increasing the sample size now that we have the system that it's up and running in the lab. So to finalize, what I wanted to present today is that our lab is studying mechanobiology across ovarian scale. So we try to link tissue mechanics, follicle development, and oocyte quality. We have developed this system to perform a high-resolution multi-scale mapping of the ovary with mechanics, structure, and function. We are also developing or using systems in vitro systems to know how the microenvironment's mechanical properties affect follicle development and oocyte quality. And finally, we are also doing some more translational work with our REI department at WashU, where we use biomechanics and AI to develop a cost-effective approach to maximize the success rates for ART patients. And with that, I just want to thank the lab that are the ones that are doing all the job and helping advance our science, the funding agencies, and special thank you to SSR for this opportunity.
Yeah. Well, great talk. We have a bunch of questions that have been like rolling in, but for the interest of time, I'll try to combine a couple of them. We'll go with one question, and I'll just remind the attendees that if you would like to reach out to foreigners, you can do that by email, and she'll also be provided a list of the questions so that she'll see all the ones that you guys have asked. So one thing that seems to be a theme through some of the questions is how are different stages of the follicles impacted by stiffness, and does this also impact steroid hormone metabolism or production?
Yeah, definitely. So we start with the secondary follicles, early secondary follicles, because it's right before this expansion is happening. So that's why we choose this one. We think that different follicles are going to be stages impacted differently. And this is something that we are really interested in exploring in the lab, if like one stage is more resistant to the other stage. So we still don't have the answer, but my hypothesis will be that yes, different stages will respond different to the mechanical cues. And with metabolism, that's something very interesting because we saw that the follicles cultured in the 0.5%, they express a lot of genes related to metabolism because they need to grow. But suddenly these genes are significantly also downregulated in the follicles cultured in the 2%. So definitely there is like all the genes related to growing are also dysregulated with the 2%.
Well, thank you. And like I said, there's a lot of questions, so we'll forward those to you. So obviously people are very engaged. For sake of time, so thank you for the great presentation. We're going to move on to our next speaker, who is Dr. Kara McKinley. She is an assistant professor in the Department of Stem Cell and Regenerative Biology at Harvard and a Freeman Hrabowski Scholar of Howard Hughes Medical Institute. Her research is focused on uterine biology and the mechanisms that regulate menstruation and endometrial regeneration. So, Dr. McKinley, the floor is yours. We look forward to your talk.
Awesome. Thank you. I'm managing to fail at the one thing we're all supposed to be able to do, which is to start to share a slide, but I think I can pull it off. Let's see. Hang on one second. It was going so well. Okay. Share, webinar, share. Okay. All right. So I'm so excited to be here and to follow Farner's that beautiful work on the ovary. We're going to shift over to the uterus because the goal of my lab is to really improve uterine health with a particular focus on the biology of menstruation. So we are focused on the lining of the uterus, which is called the endometrium. And what we are particularly fascinated by is that this tissue every month basically self-destructs. So huge proportions of this tissue are degraded and shed during menstruation, and then it grows back again. And this happens perfectly over and over and over again throughout the reproductive lifespan, month after month for somewhere around 35 years. So we think it's a really unique system to understand and harness regenerative capacity. Other systems regenerate when something external hurts them. But for the endometrium, regeneration is programmed into its physiology. It undergoes essentially a programmed trauma every month, followed by regeneration to set the system back to normal. So what I'm going to talk to you a little bit about today is what we think is at the core question around this, which is how is this incredible regenerative capacity controlled? How does it work? How does the tissue regenerate so effectively month after month and so perfectly without scarring? So this is a question that has both fundamental interest, sort of curiosity-based interest, but also clinical relevance, because the incredible regenerative capacity of the endometrium can be compromised. So while under normal circumstances, the endometrium can regenerate about 400 times over the reproductive lifespan, surgical instrumentation, for example, a DNC around a miscarriage or abortion, can compromise this regenerative capacity and result in scarring. And here I'm just schematizing for you intrauterine adhesions. So scars that are so big that they span the uterine cavity. And as you can see, essentially obliterate access to the cavity. And as a result, can cause really devastating infertility. So one of the things we might want to do to help these patients is to reactivate the regenerative potential of this system. So either activate the regenerative cells that are there, but maybe latent, or do some kind of cell therapy where we would replace those regenerative cells. To be able to do this, we need to know the answer to a pretty important question. Which cells? So that is what I'm going to talk to you about today. And this has been work from my first graduate student, Claire Eng, who is just absolute superstar, best first grad student I could possibly ask for. And then our lovely moderator, Andrew Keelaher, who has been an incredible collaborator on this project. So to understand how this process works, first we need to orient ourselves to the anatomy. So the mouse uterus, as maybe you know, has two horns, and they're basically just two tubes. So if we take a cross section of one of these tubes and look at a 200 micron thick section here, I'm showing you the myometrium, the outer muscle layer in magenta, and then the epithelium is labeled in cyan. So you can see that it has this beautiful sort of smooth structure that lines the lumen, and then it branches off into these sort of grape-like structures, which are the glands. So if I just cartoon this out for you, basically what I want you to focus on here is that the endometrium is lined by a single layer of epithelium, but there are really two types of epithelium at play here. So there's the glandular cells, which are responsible for making secretions that support a pregnancy, and then there are the luminal cells, which are the cells to which the blastocyst will actually attach. So we have contiguous epithelia, but they're transcriptionally and functionally distinct. Now, in addition to having these two very important functions as pertains to pregnancy, it's been proposed for a long time that progenitor cells also live within the base of these glands. And I think one of the things that is sort of appealing about this, I was trained actually in intestinal regeneration, and this is how the intestine works, that you have stem cells, epithelial stem cells that live in the base of the crypts that are responsible for repopulating all of the other cells that are in the intestine, including the upper villi. So the model is essentially something like this, that if we were to label permanently glandular cells and their progeny, we would be generating luminal epithelium from those glandular cells. So what was fascinating about this is that Andrew's lab made an incredibly cool mouse, which is a CXCL15 Cree, which labels the glandular cells. So this is very exciting. We can now really test this model with lineage tracing in mice. And what is super cool about this animal is that if you look at, say, a 9-week-old mouse, you look at its uterus, it looks like this. So if you don't like lineage tracing, don't worry. The basic idea is that any glandular cell or any cell derived from a glandular cell is going to be green or yellow, and any cell that isn't gland derived is going to be blue. So interestingly, we look at an adult uterus and we see yellow gland cells and not yellow luminal cells. So if I just show you the glands here, and here I cartoon it out, and then I just show you the yellow channel, you can see that we haven't labeled the luminal epithelial cells. So this is interesting because this is in contrast to the model we would have expected, the data we would have expected given our model, which is that the luminal epithelial cells would be derived from those glandular cells and therefore green. So we get to say one of the most important and like impactful phrases in biology, which is, that's confusing. And we get the same results at 9 weeks or 20 weeks. So it's not like over time we start to get population of the luminal compartment. So we started to wonder, well, maybe it's just that the system needs to be challenged. In many stem cell systems, including the intestine, the skin, other places like that, there are sort of reserve or facultative stem cells that are normally quiescent or just not participating in repair. But if you damage the tissue, it will basically activate their progenitor potential and allow them to give rise to cell types that they normally wouldn't. So basically, we wanted to know, do these gland cells start to get involved if we injure the tissue? So to do this, we developed a new approach to basically ablate the luminal epithelium. So this is an injection of basically a detergent, a surfactant, into the lumen of the uterus. So here I'm just showing you a normal, happy, healthy mouse uterus. You can see it has a luminal epithelium. But then if we do this ablation procedure, you can see the luminal epithelium is gone. But like I've told you, the mouse uterus is a great regenerator. So after this process is complete, 72 hours later, you have your luminal epithelium back. Looks great. We're happy. And what's very exciting about this is that we do, in fact, now get luminal epithelium derived from the glands. So now we have that beautiful greenish yellow luminal epithelium telling us that it was derived from those glandular structures upon this chemical ablation. So this is really telling us that the endometrium has two choices of progenitors. It can either use luminal cells use the luminal epithelium as a progenitor, so basically regenerate luminal epithelium from itself. And that's what's happening in homeostasis. But we can also make luminal cells from gland cells, and that's what's happening upon our chemical injury. So this is great. Now we know that there are separate luminal and glandular compartments during homeostasis, but that we can get movement between these compartments if we ablate one of them. So then the question becomes, what about natural injuries? I spent this time at the beginning telling you how cool the endometrium is. And one of the core elements of that is that it undergoes natural injury, actually not just in menstruation, but in both menstruation and during pregnancy and parturition. So during menstruation, we get the sort of piecemeal shedding of the endometrium. And then during pregnancy, we have a lot of disruption of the endometrium that has to happen during the pregnancy itself. And then the tissue is shed at parturition and later on as lochia. So let's just first ask this question about which of these two progenitor populations is at play during menstruation. And the expectation in the field is really that it is these gland-based progenitor models. And we thought that makes sense because the process of menstruation we think of as essentially being the generation of these sort of beheaded glands that basically during menstruation, the inner portion of the endometrium sheds, leaving behind this denuded stroma and these gland stumps, which looks pretty similar to our surfactant ablated tissue. And remember, in that context, the injury evoked gland contributions. When we did this experiment with the beautiful mouse that Andrew made, we did not see what we expected to see. So in contrast to what I proposed to you and what I showed you in the polydocanol model, we do not see green luminal epithelial cells when we induce menstruation in these animals. Instead, the luminal epithelium is still non-gland derived. So again, we get to say our favorite phrase. What's up with this? And this is now super confusing because the gland progenitor model had really made a lot of sense in terms of how we thought menstruation worked. So the basic idea, as I sort of outlined to you, but can now schematize a little bit better, was that the process of menstruation basically pops out the center of the endometrium. So you basically just have this inner layer of the endometrium, it's called the functionalis, and it is shed during menstruation, leaving behind this denuded surface dotted with these beheaded gland stumps that then resurface what was lost. But what Claire sees instead, as she goes through a very precise time course of induced menstruation in the mouse, is quite different and I think a lot more interesting. So shortly after inducing menstruation, this is what the tissue looks like. And you can see that it almost looks like the beheading happened, but it hasn't because there's still tissue present in the center here. The glands have been pushed out to the edge. And you can see that there's actually a lot of persistent luminal epithelium. That lumen has actually just been pushed to the side of the tissue, to the edge of the decidua, the edge of the tissue that's going to be shed. So we see these structures in lots of different parts of the uterus and in most all of the mice, and in different mouse models of menstruation. And we are calling them smiles because it looks like a little smiling man. And what happens over time is that we see the smile grow. So it starts to envelop more and more of the decidua, the tissue that is destined to be shed. So much so that in some samples, we can even see the tissue fully resurfaced with the shed tissue not yet expelled. But it's now intuitive that now if you pop out the center of this, you can get back a tissue that looks exactly the same as the tissue you started with luminal epithelium derived from luminal epithelium. And the gland cells have just been hanging out, moving in and out as the tissue compresses them and then returns. What's really, I think, interesting about this is that this is a way that you can shed massive amounts of tissue without exposing a lot of stromal cells. So, in many organs, aberrant activation of fibroblasts really is associated with scarring. And so, we think this is a mechanism by which the endometrium basically preempts the shedding by reepithelializing the tissue as it goes through the initial detachment process, such that very little fibroblast exposure actually happens during this process. So I thought this was a pretty cool discovery all on its own. But Claire had more up her sleeve. So one thing that she saw is that she was sort of looked at these smiles and she said, I think I've seen this before. And the reason for that is that she, a couple years ago, wrote this really beautiful review on regeneration and fibrosis in the endometrium. And in the process of preparing that review, had read, I think, sort of an esoteric paper from the 1980s and from Welsh and Enders. And that paper had a diagram that looked like this. And she was like, I think this is a smile. I think they're seeing what I'm seeing. And what is very exciting about this is that this is actually rat gestation. This is what's happening during rat pregnancy. So Claire started to wonder, to what extent is this smile process at play during pregnancy? And indeed, she sees these beautiful epithelial smiles surrounding the embryo during multiple different stages of pregnancy. And we've confirmed that these are maternally derived structures and not fetally derived structures. So this leads to massive luminal epithelial expansion during the pregnancy. And what that means is that by the time the pregnancy is delivered, you have a lot of luminal epithelium left. And actually, in the mouse, the detachment site is pretty small. So only a small amount of the tissue is actually denuded during parturition, leaving behind a very small amount of stromal exposure that needs to be resurfaced. And what she found is that resurfacing is again being driven by these luminal progenitors. So she's getting really beautiful recovery and repair in just a couple days, driven by these luminal progenitors without any involvement of the glands. So last question. I kind of breezed by this, but I kept saying that we induced menstruation. You may know that mice do not naturally menstruate. And so we have been using established protocols established both by us and by others as our ways of inducing menstruation in this system. But you may be wondering, how does this come into play during animals that menstruate naturally? So there are a very small number of mammals menstruate, about less than 2% of mammals menstruate. And it seems to have been 4 separate evolutionary events. So we have the Afrotherians, the elephant shrews, some bats, one rodent called the spiny mouse, and then the primates, including us. And we are very excited because we actually see some hints in the literature that this luminal epithelialization model may be at play across multiple of the different clades of this phylogeny. So in the spiny mouse context, a very exciting paper came out recently, which essentially said that spiny mice don't seem to have endometrial glands, which is something that we have also noticed in our colony of spiny mice in our lab. So we're very happy to know that this is a widespread phenomenon among different spiny mouse colonies because it's very weird. Glands are very important for many things. But this does suggest that if you don't have a gland compartment, you're not going to have a gland progenitor to be able to do your post-menstrual regeneration. And so this suggests that a luminal epithelium dominant model is going to have to be important in the spiny mouse. And that's something we're interested in following up. Rarely does the elephant shrew get discussed when we talk about menstruation because it's one weird elephant shrew. It's not the one that breeds in zoos. But I got one of the great slacks from Claire saying, losing my mind at these elephant shrew menstruation drawings from 1954, because she had gone back to the one set of studies that was done on elephant shrew menstruation in the wild. And the pictures look like this, which is clearly a smile. So at the time, this was referred to as a polyp. And as you sort of inherit that language without looking at the images, you lose this idea. But looking back at the images, you can see this beautiful double layered epithelium resurfacing both the tissue that's destined to be shed and the tissue that will be retained, really looking similar to our approximation to the smiles she sees in the mouse model of menstruation. So this is really starting to suggest to us that the luminal epithelium dominant models may be at play across multiple menstruating species that are separated by about 100 million years of evolution. What about primates? Everyone's favorite primate, the human. We don't know. But we have, I think there's very compelling evidence from various aspects of human data that you do get outgrowths from glandular cells during human menstruation. And there's evidence that luminal epithelium persists. So some of what we think that may be relevant from our mouse model to the human model is this idea that you have access to two progenitors, that you can use luminal cells if you have them. And if you don't have luminal cells, you're going to go for a glandular progenitor. You're going to basically deploy that backup progenitor. So we saw that we had luminal cells making luminal cells in the context of minimal luminal epithelial loss, what's happening during homeostasis, induced menstruation, pregnancy, and parturition. And then in cases of extensive luminal epithelial loss, our massive luminal ablation paradigm, then you deploy this backup progenitor. So what we think this is telling us is that there are two progenitors available that may switch in their activation based on what is available. So we think that the luminal epithelium will be preferred in contexts where it's present, but in contexts where it's not present, say in the context of massive regions of shedding during human during human menstruation, or during human parturition, which actually completely disrupts the luminal epithelium, then you might be calling into action those gland progenitors. Okay, so what is your take home? Only one take home. Endometrial regeneration proceeds with a smile. So you can see that during both pregnancy and during menstruation, these smile structures that develop from the luminal epithelium resurface the tissue, allowing for massive tissue excision with minimal exposure of those sensitive mesenchymal the mesenchymal tissues. And we actually just pre-printed this. It came out late last night / early this morning. So I really hope that you'll be able to give us your feedback here, but also take a look at the full manuscript. And please send me your feedback on that as well. And with that, I just want to thank my wonderful lab, especially Claire, who, as I mentioned, was my first graduate student. So she has seen me through the many travails of being a new PI, and really carried the vision for our lab forward with such grace and passion and creativity and kindness, which are also attributes shared by all of my lab members. And I'm so grateful to them. And of course, to Andrew and to Danvika in his lab, who made this project possible. I have been really incredible partners through developing all of this work. Here I have two preprints for you. So the Smiles and Endometrial Regeneration I told you about. I didn't have time to tell you about today about the work we've done to develop transgenic mice that can menstruate on demand. But that work is also pre-printed, and these are the two first manuscripts from my lab, so I would really love to hear your feedback. Thank you so much, and I would love to answer any questions.
Oh, well, great talk, Kara. I think both your lab and Farner's lab are building really important models for the community to use in reproductive biology. We have a bunch of questions now rolling in. So some of them are really interested in how you induced menstruation in mice and the different models you may have used. So if you can comment on that.
Absolutely. So if I really reduce it to sort of simplest terms, the distinction between most menstruating species and non-menstruating species relates to their regulation of a process called decidualization. It's basically a differentiation event that happens in the endometrium. And if you decidualize your endometrium during your secretory phase, during your progesterone high phase, when the progesterone falls, you get menstruation. So the classical models have actually discovered that you can induce this decidualization event in a non-pregnant mouse in its progesterone high phase by injecting oil into the lumen of the uterus. I find this whole, I'd love to hear a personal narrative of how this was discovered. And it's very effective. And we did that in a lot of the lineage tracing experiments that I showed today, because it is beautifully compatible with all the super cool mice that Andrew made. The other model that we developed uses information that has been sort of actually floating around in the field for a while about the molecular inducers of decidualization, which taught us that increasing cyclic AMP is a major determinant of decidualization. So what we did is we borrowed a tool from the neuroscientists. They have these modified GPCRs, these engineered GPCRs called DREADs, which drive certain signaling pathways when you give them a defined agonist. So one of them is called a cyclic AMP DREAD, GS DREAD. And so if you give that the agonist, you get an increase in intracellular cyclic AMP. And doing that during the progesterone high phase of the cycle is also sufficient to induce decidualization. And when the progesterone falls, menstruation. So basically, two mouse models of menstruation, one that uses oil as the decidualization stimulus, and the other that uses this transgenic receptor as the decidualization stimulus.
Um, so that was a great answer. One of the questions I think that popped out to me on here that I'll bring up is, how do you think regeneration is impacted in women that have fibroids or other uterine diseases?
That is a great question. And we are very fascinated by this now. I think there's lots of different contexts. The short answer is I have no idea. Um, and some of that may affect intrinsic factors, extrinsic factors to the endometrium. We're very interested, for example, in my lab in heavy menstrual bleeding and the role for deviations in repair in leading to excessive blood loss. And I think one of the things we're really excited about is now looking at all these models where people have previously found defective repair and asking what part of the repair process that we've now sort of characterized in more detail is defective in those contexts. So basically, can you not make a smile? Can the smile not grow? There's actually beautiful work from Liz Robertson's lab. Um, it's a Nature Communications paper on BLIMP1. And basically, what they found is that BLIMP1 knockout animals don't shed their decidui, and they also don't make smiles. So we're very curious about whether smiles and shedding are coupled in that way.
Yeah, I think we have time for one more. So again, I'm trying to kind of amalgamate a couple of these together. People are interested in stem cells. So I think based on your background in the gut, do you think that there's separate luminal or glandular stem cells, and is there a role for classical markers like LGR5 in this regeneration?
Um, so I don't think I can I don't think I have data to speak to that necessarily. I think there's nice work from Pradeep Tanwar's lab showing that axin 2 positive cells in the gland base regenerate the upper part of the glands. So I think one of the things that's been a little bit confusing and was confusing to me until I overthought it constantly is what does it mean to be a progenitor and how do we assess that? Because say a gland-based progenitor needs to do two things. Yes, it needs to regenerate the luminal epithelium. That is what I have talked to you about. But it also needs to regenerate the upper gland, which is also shed during menstruation. And I think we often conflate those. So there's actually really cool data in humans where they use rare somatic mutations to trace clones, and they show that gland-based cells give rise to upper gland cells. So there's clearly progenitor potential in the base of glands that is sufficient to make upper glands. Whether it's sufficient to make luminal epithelium, I think is not clear in humans, and I think our data in mice would argue mostly no unless there's an ablation. So I also think some of these things where we're studying progenitor potential based on sort of self-renewal or the capacity to make organoids, things like that, they're actually really testing the capacity to make gland cells, which is itself a progenitor, but a more restricted one than what would make luminal epithelium.
Good job navigating those questions.
As deep in the weeds on the stem cell stuff there, sorry.
Yeah. Okay. Well, I think we're coming up to time right now. So I just want to take the opportunity to thank both of our speakers again. I think this is really highlighting some really up and coming faculty in the field doing really exciting work. So I want to thank everyone in the audience for joining. Of course, we'll pass your questions along to our two speakers and they can answer. Um, and then, so with that, I guess we'll conclude today's webinar, and we hope to see everyone both at SSR and other SSR virtual events. So thank you, everybody. We had great turnout today.
Thank you.
Thank you.